Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Metabolon Incorporated, I'd like to welcome you to Harnessing the True Power of Metabolomics with the 4 C's. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now, I'd like to introduce our presenters for today. First is Gregory Michelotti, PhD, Associate Director for Discovery and Translational Sciences with Metabolon. Next, Annie Evans, PhD, Director of Research and Development for Metabolon. And our third presenter is Kirk Beebe, PhD, Senior Director, Discovery and Translational Sciences with Metabolon. Now, I'll turn the webinar over to our presenter.
Thank you for the introduction, and I would like to first start by level setting of what metabolism is and to help understand what metabolomics and why we think metabolomics is the key to understanding biology, health, and disease. Metabolism is the conversion of food and fuel to energy to run all cellular processes, and these enzyme-catalyzed reactions allow organisms to grow and reproduce to essentially maintain the biology of life. And it's the intermediary and chemical endpoints of these enzyme-catalyzed reactions that are called metabolites, or biochemicals, that I will use interchangeably. Metabolomics is the large-scale study of these metabolites within all biosamples, which include cells, biofluids, tissues, and organisms. And metabolomics represents the quantitative assessment of these changes in these biochemicals to understand how they influence biological systems. And metabolomics is really the keystone of systems biology in that it integrates both genetic and non-genetic influences to provide a definitive representation of a phenotype. And on the top part of the slide, shown in purple, are what is typically thought of the flow of genetic information, or the central dogma of molecular biology, that DNA goes to RNA goes to proteins. And what I like to point out is that metabolomics integrate this information from these upstream modalities to integrate both this information as well as the diet, lifestyle, microbiome information on the bottom part of the slide shown in blue. But on the top part of the slide, what I like to say is that there's a level of complexity with these omics modalities that confounds our ability to understand how they operate across different model systems, for example, from yeast to mice to humans, and even within individuals, such that genetics, for example, they display a wide range of genetic variations. Transcripts, RNA, go through a wide array of post-transcriptional modifications, such as splicing and processing, and proteins undergo a bewildering array of modifications post-translationally, myristylation, phosphorylation, nitrosylation, etc., that confound our ability to understand how these operate in different model systems. Unlike metabolites that are conserved from bacteria to humans, and that structural conservation implies functional conservation, such that the TCA cycle, for example, is conserved in bacteria as it is in humans, and that helps us understand how they work across different model systems. But also, it's the integration of xenobiotics, exposure-related metabolites and biochemicals, microbiome, and lifestyle that makes metabolism and metabolomics a really unique modality to understand how these are all integrated to provide a comprehensive view of a phenotype. Microbiome, for example, is a buzzword these days in that bacteria and bacterial-produced metabolites influence a wide variety, almost every variety, of human health and wellness and transition to disease. And we're able to capture this information, as I said, to provide a comprehensive view of a phenotype. But that phenotype has a research abstract in that what we really want to understand is real-world outcomes. And metabolomics is the key to driving optimum insight from all these upstream omic modalities, as well as external factors such as exposure, microbiome, and lifestyle, to provide real-world outcomes for what's going on in an individual, such that we can functionally map upstream omics modalities such as genetics to understand what's going on in genes and genetic variants and how they influence health and wellness and real-world clinical outcomes, and all these other non-genetic influences that also affect, like the microbiome, that affect health and wellness in a real-world status. And now I'd like to provide an example of why we feel metabolomics provides really unparalleled insights that are unseen with genomics. And this is an example of sickle cell anemia, which is still a highly prevalent disease today. For example, in Africa alone, in Nigeria, 150,000 children are affected with this on a yearly basis, and over 15,000 individuals, patients, die worldwide as a result of this disease. And this is despite knowing the molecular basis of this disease has been known for over 60 years. It was in 1956 that Vernon Ingram identified a mutation in the beta globin gene that changes a glutamine to a valine at position 6. And this induces conformational changes in the beta globin that induce a change in the red blood cell shape, so that it produces a sickling shape, which is shown on the bottom part of that slide. And despite knowing the mutation for over 60 years and the genetic basis of this disease remains untreatable to this very day, and the mechanistic basis of this disease are unknown. And it wasn't until a metabolomic study was done recently, flash forward 60 years, that insight was provided into the metabolic basis of this disease that provides therapeutic opportunities and insights for how to treat these individuals that are currently facing an untreatable situation. Metabolomic profiling identified perturbations in a number of pathways, including adenosine, lysophospholipid or fatty acid metabolism, 2,3-diphosphoglycerate, as well as amino acid metabolism, shown here aspartate on the right part of the slide, that showed that 4 major pathways are perturbed relative to control individuals that are shown on the left part of that panel, such that this provides therapeutic opportunities for these individuals. For example, adenosine at the bottom part of that panel is a receptor-mediated system, and being receptor-mediated system, this provides therapeutic options for drug targets to go after. And in fact, this is under review today. So it's metabolomic profiling that provided unique and powerful insights to new therapeutic opportunities to treat patients that had been essentially clinically left behind for over 60 years. And it's not just diseases in large populations where we see the power of metabolomics. It's also in understanding rare diseases and ushering in the era of precision medicine. For example, we did a collaboration recently with ARPODET at Baylor University, where we showed what the power of metabolomics can be used for identifying rare disease and inherited metabolic disorders in children's samples or pediatric samples. And in this collaboration, we analyzed 200 samples from patients that were sent to us in a blinded fashion. Some were defined with disease, undefined disease, they were sent to us in a blinded fashion, and some were unaffected. And the challenge platform to determine who's affected and who's unaffected, and correctly to identify the cases of disease. And the results were that we identified the cases with a high degree of precision to correctly call all these patients. And this really shows the power of using this approach to identify rare metabolic diseases in individuals, such that we developed a single test that can identify the potential for over 60 rare congenital metabolic diseases from a single patient sample. And this kind of approach reduces the cost and time of diagnosis and identification for the best course of treatment for these patients. And this can also be used for the longitudinal monitoring and assessment for treatment efficacy, because again, metabolomics represents the real world outcome and actionable information of what's going on in an individual. And I'd like to point out that every disease is a metabolic disease, that any change from a homeostatic step to a disease step requires a change in a biochemical step, and that can be detected through metabolomic profiling, so that a wide array of diseases can be affected. For example, neurogenic diseases or personality disorders are at their core also metabolic disorders. And this is an example of epilepsy. And there are patients with refractory epilepsy who display a wide range of clinical manifestations that are shown on the left part of that slide. And it's been known for a while that a ketogenic diet is effective in the treatment for refractory epilepsy, but it's a mystery of how it worked. So this is a case where diet is driving changes in a disease state and used for treatment, and really represents an ideal situation to view metabolic profiling. So through metabolic profiling, it was known that the ketogenic diet changes the microbiome composition, and that this microbiome composition changes metabolic profiles in individuals. And the specific metabolic step that was changed was a change in gamma-glutamyl amino acids that resulted or alters the change in the GABA to glutamate ratio in individuals, so that through metabolic profiling it was found that this ratio is disturbed in epileptic patients, and that through ketogenic diet this provides a beneficial change in glutamate metabolism and gamma-glutamyl amino acid metabolism, so that it alters or increases the GABA to glutamate ratio in the brain, producing beneficial responses. And in fact, this is the basis of a current treatment option to inhibit gamma-glutamyl amino acids because it's an enzyme. It represents a drug target. So through metabolic profiling, this is the identification of a pathway and a drug target that have proven effective for the treatment of refractory epilepsy. And none of this can be realized without the core competencies that drive metabolomic understanding and insights forward. And now I'm going to turn it over to my colleague, Annie Evans, who's going to discuss these core competencies with you and how it's important for driving biological insight.
Thank you, Greg. We've just seen incredible case studies where metabolomics is used to drive real insight into a variety of different areas of research. But you can't just have any metabolomics technology. What we know is that there are really some core elements or capabilities that allow this process to become important. And these insights could be actually different for any given researcher. It could be somebody looking to identify a biomarker for a specific disease or to be used as a companion diagnostic. It may be an understanding of the disease progression or mechanism of action of treatment. So the insight that you're looking to derive could be very different depending on the type of research, but is really the combination of these four elements which come together to give you the greatest potential for making those biological derived insights. So these four C's are coverage, competency, comparability, and capacity. And over the next few slides, I'm going to talk about these with greater detail. The first C is coverage. The definition of coverage is the ability to see and compare thousands of metabolites to reveal new insights and opportunities across diverse markets. Coverage is really what we've been talking about, that you have to have the breadth of the number of molecules detected and the number of different biological processes being covered. Greg showed us that it's not just about the endogenous processes, it is also the influence of these external factors, like whether you're taking over-the-counter medication or what is the state of your microbiome. So if you look at metabolism through this keyhole, you are going to have a very difficult time making a really strong biological assumption or driving much biological insight. I use the analogy of making a decision about whether or not you're going to take your family to a picnic today based on this blue sky picture on the lower left-hand side. And you might look at that and say, yes, absolutely, let's go for a picnic. Or on a biological side, if you're looking to see if a patient is progressing towards a disease state, you're going to look at this blue sky picture and say, no, this patient is perfectly healthy. The problem is when you have the complete landscape, what you see is that, in fact, you know what, maybe no picnic today. And then, in fact, your patient is having markers of early stage resistance or early stage maybe toxicity profile to particular drug treatment. You only see this when you look at the entire landscape and really get a complete coverage of all metabolism. Now, Greg talked earlier a little bit about the importance of microbiome. We know that this is a particularly interesting area where landscape and viewing the entire system is important. Most other omics systems really don't have any ability to understand the influence of the microbiome on the host, and understanding that biochemical relationship is really key to understanding microbial research. The next C is competency. And competency is defined as the ability to generate high-quality data driving biological insights and making actionable recommendations. I think of this as being essentially the core of any of your technology that you're going to use, all the way from collaborative study design all the way through the process of actually doing the interpretation of the data. You have to have deep understanding of your entire process in order for competency to be there. So this means having a technology which has quality regulatory systems in place to make sure that the data that's being produced is high confidence and high quality, making sure that you have a pipeline of informatics that allows you to generate statistics which are both accurate and can be relied on, that aren't going to generate false discoveries. And no small part of this is the interpretation of the data. You have to have the tools and the expertise to be able to make sense of the data. There is a real danger within the metabolomics technology to send this very large list of molecules, some that are going up, some that are going down, handing it to an investigator and saying, here you go, good luck, and having that investigator not knowing how to analyze the data, or worse yet, not having the tools to analyze that data, and so putting metabolomics data in the drawer, shutting it and saying, you know what, metabolomics wasn't able to help me. The real power here in competency is knowing what the signatures are of each of these biochemicals, knowing that when those three molecules are going up and fatty acids are going down, that you're seeing a disruption in lipid metabolism, or perhaps those are signatures of liver malfunction. They all have specific annotations and signatures, and that is a really important part of competency, is knowing what these signals are and how they relate to these specific diseases. The next C is comparability. Comparability is defined as the ability to compare the data from different researchers in different geographies over time, but also among different patient populations, races, and matrices. Now, Greg talked earlier about the importance of precision medicine. One of the places where comparability becomes incredibly important is as we move metabolomics into this very exciting area of precision medicine, i.e., as Greg described before, the ability to analyze a patient as an individual and compare them to a healthy population. Every time we want to run an individual infant sample, I can't analyze the healthy infant population at the same time. I have to be able to compare that infant today to a healthy population which was potentially run over 5 years ago. To be able to do that, you have to have a firm basis in the technology to compare the data collected at very disparate time points along the journey. And so comparability is this ability within a technology to make those leaps. The other particular interesting area where comparability is really important is as we move into these much larger population data sets, potentially thinking about longitudinal studies where you are tracking individuals over the course of their lifetime. You don't want to have to wait until 80 years and all the samples are collected to start looking and driving insights along that path. You want to be able to compare and merge that data as you go. But even bigger than that, you want to be able to compare your aging cohort to that population cohort in the UK, to maybe somebody's diabetes cohort that was run in Finland, right? You want to be able to take your data and have a data standard to be able to compare your data to other people's data. And that is something that has to be present in metabolomics technology to really push this forward and to have it be incredibly impactful for deriving insights. The last C is capacity. Capacity is the ability to process hundreds of thousands of samples quickly and cost effectively to serve rapid growing demand. Ultimately, capacity boils down to how quickly you can get your data back, right? Metabolomics data and the analysis of the data need to be attainable within a reasonable amount of time so that it can support the needs of the investigator. We all know, we've all been there, projects change rapidly, priorities shift really fast. And if we do not have data back in a reasonable amount of time, you're not going to be able to make the appropriate decisions in terms of your pipeline, or am I going to move this candidate forward or not? So if you have lengthy and undefined turnaround times, this can be really detrimental to the progress. So capacity really speaks to the ability to have a defined project timeline, as well as know that you're going to be able to have a refined process that you can use over and over again. So I've described now some of the 4 C's and how they relate and how these are foundational to being able to drive metabolomics data that are ultimately going to be the most profoundly impactful to the data. Now, my colleague Kirk Beebe is going to be talking to you about some of the case studies that we've done where metabolomics, as enabled by the 4 C's, has been implemented to really find meaningful data within the framework of drug development as well as the management of product life cycle.
Thank you, Annie. And as Annie mentioned, one of the areas where there's been a lot of application and also impact using this approach she just described is across pharma R&D. And really, this extends from very early discovery all the way deep into clinical studies. But I'm going to actually dive into a couple areas where there's a pretty high frequency in terms of activity, and it's described on the next slide. And if we go back to some of the lessons that Greg was conveying in his intro about what metabolomics can do, essentially it can offer a fingerprint of the phenotype or the physiological trait of a system. Really what people are ultimately doing with pharma R&D with metabolomics is they're using it to get really clear and relevant signatures, ultimately of that target molecule combination and then those associated biomarkers. And what I've highlighted on this slide over on the left is really where that starts in animal models. And ultimately, the idea with that is dosing your compound target combination to be able to see essentially, are you hitting the target? Are you seeing a signal that reflects efficacy? Are you seeing any potential off-target effects or liabilities? And by doing that in kind of one shot with a metabolomics study, you can gain a really deep understanding of the MOA and the target molecule combination. And this is really highly relevant for both large pharma as well as small and emerging pharma. The other thing about that, besides just that deep insight that you get from that, is you actually also, those same signals of hitting the target, being able to see pathways reflecting efficacy, and then potential liabilities, those same signals can be converted over to biomarkers as well. And that's kind of the subject of the second part of the slide here on the far right. That is trying to use this technology, as Greg also mentioned, the translatability of metabolomics, with model systems overlaid with appropriate human cohorts to find translational markers, signals, to evaluate your models, to make sure that they're as close to the actual human case as possible. And one thing about that that second part, the translational studies, is these can be done sequentially, but I think ideally they're done somewhat concurrently. That is, and when you're in the model systems, you're looking at relevant cohorts, or maybe even it can also overlap nicely with, for example, phase 0 studies. So the examples I'm going to take you through actually reflect these cases here on this slide. So the first example is actually of that widespread molecular profiling or molecular phenotyping of a rodent model. And this was actually an instance where a large pharma actually had a compound directed against a novel target that was that was causing acute renal failure in mice. And they knew the toxicity wasn't related to the mechanism of action based on a knockout, so it was clearly off target. And what they had essentially done is they exhausted every other kind of profiling or standard method. So they actually sought to see if metabolomics could actually find a marker or a signature that can inform on this, and more importantly, predict this occurrence. And shown on the bottom here is really just a schematic of the study, a very condensed schematic. But basically, just simply did a dose escalation study, acute dose escalation study. And what you're seeing here are just a z-scores of metabolite changes across the study. On the bottom in red, you actually are looking at the high dose, and that's where the acute renal failure is occurring. But zooming in on a number of those changes in this plot in the middle, you can see one particular metabolite that was really important in this study. It's in the red trend line here with the high dose once again, and this is 3-indoxyl sulfate. So this is going up very quickly after dosing, and it comes down slowly after dosing. Importantly, if you look on the bottom in kind of the blue trend line, you see other markers, urea and creatinine, that are more traditional established markers, and you can see why they were having trouble. There's really not much of an effect with those. So with this finding, they actually were able to functionalize this finding in a practical way by developing a targeted assay of just this molecule and then screening other chemotypes. And shown on the graph on the right here is the results of that, where you can see the original compound in red that was showing the toxic effects. You can still see really high levels of this molecule, 3-indoxyl sulfate, but with other chemotypes, you see that that effect isn't quite present. So this really gives them a practical way to use this initial finding. But initially, it was really finding a more sensitive biomarker than what are the traditionally used. That was really key. This marker actually has a lot of translatability as well. And then lastly, I think this is a clear illustration of how this could help the immediate program directed against this target, but it also is something that could be added across programs as just a screen for other programs in this toxicity event. So the next example is actually more about the finding targets and also translational biomarkers that are more related to efficacy, in contrast to the previous example, which was related to safety. So this is actually in autism spectrum disorder, or ASD, and this started out in a rodent model. Obviously, it's fairly well appreciated that ASD, the incidence continues to grow and therapies are limited. So this initially started with researchers at Caltech finding that a mouse model with ASD-like traits had differences in the gut microbiome. But they didn't really know how the microbiome produced the effect, how to monitor it, and the translational potential, obviously, for mouse model was unclear. So what was done is a metabolomics screen of the blood from this mouse model. And one key finding is shown here on the left in this graph. This molecule 4-EPS, you can see in the control mice is very low. In the symptomatic model in the middle, it goes up very highly. And then the treated symptomatic model, it goes down again. And it also tracks with this behavioral phenotype as well. So this is a pretty exciting finding. It gives a potential target to look at and also a biomarker, but of course it's in a mouse model, and that obviously could be a galaxy away from a relevant human condition. So actually it was really important that last year at the International Society for Autism Research, findings were presented on this particular marker in a fairly substantial human cohort that showed that it actually repeated in this particular cohort. So this appears to be a marker that has good translational potential. Clearly, it would be a valuable marker in overlaying it with a development program directed against this particular dysbiotic-driven autism. And incidentally, there are active therapeutic programs that are ongoing right now. Maybe the wider lesson here too is and I think Greg and Annie both reflected on this, is this actually continues to build on this idea that if people want to understand the microbiome, they need to look at metabolites because they're often the functional end of this interaction. And the final example I want to show is just oftentimes you can directly, as I showed on that Venn diagram on an earlier slide, you can actually profile clinical samples directly, whether it's a phase 0, phase 1, or in this case, a phase 2 study. So this is really a fairly common occurrence where investigators are looking for markers that can predict response in clinical development. This goes essentially across indication areas. This happens to be a particularly high need area of NASH, or non-alcoholic steatohepatitis, where really the circulating or blood-based biomarkers are very limiting, where biopsy and imaging as a kind of a powerful adjunct are really the staples. So they were looking for novel biomarkers in blood of subjects in this phase 2 trial with this novel inhibitor against NASH, essentially an ACC inhibitor, or acetyl-CoA carboxylase. And just to quickly summarize, they screened about 700 or 800 metabolites, and there was one cluster that actually tracked with response, so imaging response, and it's shown here in this plot. This cluster of metabolites, they happen to be acyl carnitines. At the high dose, where there was the highest prevalence of response, they actually declined nicely across multiple classes. And again, this was a metabolomic screen, but at EASL a couple months ago, back in April, it was presented that taking these markers into a targeted assay, they indeed do repeat and therefore appear to be promising early markers for this imaging response in patients on this NASH trial. The other thing that I don't really have a lot of time to get into is that these markers also very logically tied to the mechanism of action of this particular inhibitor. And then the other thing I think that is important here is that this, I think, illustrates the ability to find other signals that can build program confidence moving forward. And clearly, as they move forward, these markers can accompany future development in looking at the pharmacodynamic activity of this particular target. So those are just a snapshot of a few examples. Again, work using this technology in the manner that Annie described can be very early discovery all the way through the clinic. Before I hand off to Greg to talk about another area of pretty important application, I wanted to just transition because I think there is an area that Greg is heavily involved in that actually has really nice overlap with one area of this chevron. And that's actually in the very first part of it, which is discovering targets, making sure you know you have the right target. So there's a lot of activity in that space as well these days. And if you think about it, I'll show you on the next slide. The pursuit of new targets is really very aggressively being looked at through human genetics. Really, since this landmark paper that's on the right of this slide was published, as well as some additional follow-on work, and there's a recognition that targets that have strong genetic evidence have a higher probability of success. So therefore, you can imagine, and if you look at the popular press today or read science articles, you'll see that people are being sequenced, large cohorts are being sequenced around the planet right now to understand human health and disease. But from a pharma perspective, it's to find relevant druggable targets. And as my colleague Greg will illustrate in this last section, metabolomics has been a really key tool for finding out what are those important signals within the genome, and then also functionalizing that information to how they're driving particular meaningful phenotypes as well.
Thank you, Kirk, for that really exciting overview of how we're working with biopharma to accelerate their findings for both drug discovery and drug efficacy workflows. In this next segment, I'd like to present what combining the power of 4C metabolomic profiling with the statistical power of large human cohort studies can do to increase our understanding of both health and wellness and transition to disease. These large population cohort studies examine very large numbers of individuals to gain insight into specific questions of clinical relevance, because people in very large numbers do very predictable things. Heck, this is the basis of the entire insurance industry, and they seem to have success with this approach. As an example of this, I show two panels on the left, and this is work from Zhenming Chen at Oxford University, who showed a correlation with increased blood pressure and increased risk of ischemic heart disease and mortality. On the very left part of the panel, he studied 5,000 adults to look at this correlation between these two modalities, and found that across different age ranges, going from 40 years old to 90 years old, very loose and noisy correlations between increased blood pressure and increased risk of mortality from ischemic heart disease. In fact, to the point where in the 80 to 89-year-old group, it looks like there's a decreased risk of increased blood pressure and increased risk of mortality. And it's not until you do very large numbers that you see people doing very predictable things. So that when we expand this population 5,000 to 500,000 adults, you begin to see clear patterns of how increased blood pressure correlates with increased mortality through ischemic heart disease, to the point where there is almost a one-to-one correlation across all age groups. So that by these large population studies, we see the increased power of being able to identify risk factors for human disease. Now I want to briefly review an exciting recent study that combines cohort power with our 4C metabolomic profiling to integrate each of the areas that Annie and Kirk reviewed to provide unprecedented insight into a growing problem: high blood pressure and increased risk of mortality. In a study from Anna Valdez at King's College in London, they looked at 3 distinct cohorts, doing metabolomic profiling in these cohorts, and identified increased levels of a single metabolite, dicarboxylic fatty acid called hexadecane-dioate, that I'll abbreviate hexa for short, and found an association with increased blood pressure in these populations. And as we saw from the previous slide, this has a high degree of association or correlation with increased mortality. Aside from this association with a real-world outcome, blood pressure, they, in a parallel set of studies, identified increased levels of metabolite with variants of unknown significance, in this case, a solute transporter called SLCO1B1. So that collectively over these studies, they not only functionally mapped a gene of unknown function, a solute transporter, with levels of this gene, levels of this metabolite, they also showed that levels of this metabolite correlated with increased risk of blood pressure. So collectively, this provides a hugely clinically relevant area of how metabolomic profiling can be useful in population studies. But association is not causality. Follow-up studies by this group were preclinical models where they actually dosed animals with hexadecandiol directly relative to control groups that were treated with vehicle alone. And as can be seen by the middle panel, dosing of these animals resulted in a direct increase in systolic blood pressure relative to control animals, which is the blue line relative to the control yellow line, so that they show a causal role for dosing this metabolite to increase blood pressure. Further, parallel studies by a separate group, Bing Yu and Eric Boerwinkle, in a separate cohort, the ERIC cohort, which is largely comprised of African Americans, were looking at changes in metabolites that were associated with SNPs. They found in this population that relative to control individuals, which have the reference alleles or homozygous for no change or no mutation, that individuals with loss of function in one allele of the SLCO1B1 gene showed markedly higher levels or measurably higher levels of this metabolite relative to control individuals that had both normal copies, and that loss of function of both alleles, which is on the right part of that panel, on the right side of the screen, shows markedly higher levels of hexadecandiol. So this really helps usher in an area of functionally mapping genes and genetic variants to metabolite levels that have associations with real-world outcomes. And this is important because by over 2020, over 1.5 billion people will have high blood pressure, and 2/3 of them, close to 1 billion people, will not achieve their target therapeutic control with the current therapeutic opportunities available to them. And in these series of studies, we've provided insights into new pathways to treat drugs and present new therapeutic opportunities to treat a still growing problem in the world. And in fact, we're working with many cohorts over very distinct populations to bring the power of metabolomic profiling to population health, such that we will be able to understand in very deep detail health and wellness and transition to disease through metabolic profiling. And we're currently working with Twins UK, the Million Veterans Program, Canadian Longitudinal Study of Aging, and Direct IMI to work with them to provide this insight. And none of the cases that Kirk and I discussed earlier in the slide would be possible without the 4 C's that Annie discussed earlier, to show how the power of comprehensive profiling enables us to address leading scientific challenges at scale. And Annie now is going to discuss how Metabolon defines and delivers on these 4 C's.
So again, just to rehash what everybody has said, is there's incredible insights to be derived from metabolomics within vast areas of research. But we know that there are core competencies which are really key to be able to deliver this, and Metabolon delivers on these. Metabolon has the largest breadth of coverage of anyone in the industry. We have a known authentic standard library of over 5,200 compounds, which include the microbial-derived metabolites, common over-the-counter medications and prescription medications, but also complex lipids. This library allows us to be able to detect and identify between 2,000 and 2,500 metabolites in human plasma, so really expanding the coverage of biochemical diversity in an extremely comprehensive way. The other thing that we are always aware of is the novel discoveries which are in this data. In addition to our known library, we have a library of unknown or unnamed metabolites, and we have entire initiatives within the company to actually attempt to identify these unnamed compounds. And many of these, just to give you a hint, many of these are ultimately being metabolites that are not known to man, and are many of them believed to be microbial-derived metabolites. So it's incredibly exciting to be within a technology that also enables us to really move the cutting edge of an understanding of what metabolites are actually even present in these samples from the get-go. Metabolon has been doing precision metabolomics for over 15 years. We have a technology which is regulated, internationally regulated, that has quality control built in through the entire pipeline, Metabolon really sees competency as being more of wanting to be a partner with you through the metabolomics process, including discussions about what makes good study design, what are good statistical approaches to be done, in addition to being able to bring the insight of what does it mean when your fatty acids are going up but your branched chain amino acids are going down, as an example, bringing the sort of annotation experience with over a decade of experience of looking at metabolomics studies, and obviously covering a breadth of species and sample types, such that over this time we have over 2,000 publications, and in the last year over 12 or over 27 in high impact journals. So we offer a tremendous competency to the challenges that can be metabolomics. Now, finally, one of the things that we believe quite firmly in, or the next C, comparability. For this technology to be the most impactful to our science, we have to have comparability. Right now, most metabolomic studies are independent case control studies that have no real ability to tie information between these studies. They are all distinct and independent. For metabolomics to be the most impactful on science, we have to create a data standard, and Metabolon is committed to being and creating this data standard for metabolomics, such that anyone that runs a project with us will be able to compare, and we will be able to help them compare their data to projects run by other cohorts or run by other researchers within the community, and we will assist with those collaborations and want to help drive those interactions. So as we move forward, comparability is just going to become a bigger and bigger part of metabolomics and driving its success. The final important aspect of a successful metabolomics study is capacity. Metabolon has the world's only global metabolomics network, meaning that we have labs in multiple places around the planet where samples can be run. We have a lab in Research Triangle Park in North Carolina in the United States, in Potsdam in Germany, in Doha in Qatar, and in Shenzhen in China. Each one of these labs have their own sample capacity, but at the headquarters in Research Triangle Park, we can run over 200,000 samples a year. This means that we have the ability to support research on small pilot-level research-based projects all the way up to very large population-type research projects to be able to support you throughout your pipeline needs. Ultimately, your success is our success. And while I know that sounds cheesy, this is truly how we believe it. If we can't help deliver solutions to your scientific problem, we have not served you, and metabolomics has not served you. Metabolomics, we feel, is incredibly primed to answer some of the most difficult questions in life science research, particularly if you bring this together with some of the other multi-omics, which Greg talked about in his talk, with being able to combine some of the information you get from genomics approaches, which can be really empowered and even brought to a greater level of understanding when metabolomics is also brought into that. We are very excited to be in this research at this time, and we hope that the seminar today has given you some insight into what we believe are foundationally important aspects of metabolomics, as well as encourage you to think about how it can be used in your research. We would like to thank you very much for your time today, and I believe we'll be taking questions. Thank you so much.
Thank you all very much. Yes, we have quite a few questions, so let's get started. Our first question, did Metabolon perform the metabolomics analyses of all of these samples? And if so, which platforms were used?
Yeah, go ahead.
Hi, this is Greg Michelati. Metabolon did perform all these analyses, so this was all using our technology. And this is using an LC-MS-MS platform to analyze all these samples. It's a global platform, so we can compare and provide consistency across all the sample sets to provide this biological insights that you saw today. Thank you for your question.
Okay, and our next question is for either Annie or Greg. What sample types do you analyze, and can samples from different types be compared directly?
Yes, that's a great question, and so I'll answer that. We can look at all sample types. Basically, if there is a biological sample or biochemicals that are present in the sample, we can extract those and analyze those across our platform. So we can look at an incredibly wide swath of samples. We've looked at all biological samples, including circulating metabolites, blood plasma, urine, tissues. I think we've done fingernails, mummified tissues. So if there are biochemicals that are present in the sample, we can extract them. Unfortunately, because there are different metabolomic or biochemical compositions in different sample types, this precludes their direct analysis. But we can provide comprehensive coverage of virtually every biochemical sample or biological sample.
The one thing I will add to Greg's comment here is that we had a specific question in addition to just sort of general sample analysis about FFPE samples. This is formalin-fixed paraffin-embedded samples. There's actually a publication out there, and forgive me for not remembering off the top of my head. I believe it was with Dana-Farber, or potentially it was with the National Cancer Institute. I can't remember right now, where we actually looked at different comparison of different storages of samples, particularly looking at FFPE. And it was really interesting paper, so hopefully you can follow up with us and we can get you the link to the paper. But either way, it showed that FFPE does certainly have a significant impact on the metabolomics data, i.e. you see a lot fewer compounds in FFPE type analyses, but actually you still were able to define cancer-specific signatures even in the FFPE. So while the FFPE absolutely does have an impact, you were still able to see signatures. So I think it's an interesting discussion to have about what are the goals of the project. I don't necessarily think FFPE is great for every study, particularly if you're trying to understand mechanism of action, but if you're looking for biomarkers and diagnostic markers, then I think it could be absolutely an option for you.
Thank you very much. The questions are coming faster than I can sort them, but I'm doing my best here. For Annie, could you please discuss in more detail the statistical support that you offer?
Absolutely. So we have an entire stats department, if you will, several PhD statisticians, and ultimately what we want to be able to do is work with you so that we understand what your needs are, right? We don't have defined we have defined statistics that we like to do, but inevitably we want to work with you and say, well, depending on the impact of depending on the need of the study, perhaps hierarchical clustering is what you need, or perhaps just pure t-tests or PCA, or, ultimately we can do any of the statistical analyses that are available to you. It's just about understanding which are going to be most appropriate for the study in terms of what the project goals are. There's not one set of stats that you want to run because they're really not appropriate for every question being asked.
All right, a question for Kirk. Isn't covering more just increasing the chances of false discovery?
Yeah, that's a good question, and it's pretty common. So I'm going to actually take some liberties and say false discovery here is referring to the end point, the biology, not necessarily statistical false discovery. And I think if you go back to one of the slides I think Annie showed with the keyhole over the metabolome, there's a tendency for people to be much more hypothesis driven and think that looking at a smaller window is actually more beneficial and will prevent false biological discovery. But I think a lot of the examples you showed here today reflect that actually covering more prevents you from going down rabbit holes or finding red herrings and actually getting the signal that's relevant right away. So I actually think counterintuitively to what maybe we've been kind of taught in the omics space, seeing more in the case of metabolomics is actually better.
Excellent. And a question for either Greg or Annie. How many compounds can you identify and which pathways are represented?
Yeah, that's a great question as well. We screen for over 5,200 named molecules in every sample, so that these cover the broad range of biochemical pathways. And we also screen for another several thousand unnamed biochemicals that are recurring biological entities that occur in samples that we're able to track and quantify. So collectively, we're able to screen for over 8,000 molecules in a given biological sample. And as I said, these cover the broad range of all biochemical pathways that have been identified. So in this way, we're really able to provide comprehensive coverage of any perturbations that occur in a given study design. So we're able to provide deep insight into what the biological questions may be for investigators.
And again, just to add to that, I think it's really important to note, too, that the molecules that we're screening for include all the sort of common known endogenous pathways that are really well established within mammalian systems, as well as all those microbial-derived metabolites, which are really sort of pretty new and novel, and we don't have a real great understanding of the diversity of these molecules present in human samples or any sample type. So all of those are covered, as well as, again, dietary components. You know, some compounds you can only get from diet. You know, I always tell the story that you can joke around that if you drank orange juice for breakfast, I'm going to be able to tell that in your plasma because there's specific molecules coming from the orange juice. So it includes all of those things, and you know, over-the-counter prescriptions, common prescription drugs, because again, those things can go right towards whether in a clinical trial everybody is following the mandate of what you've asked them to do. People don't realize that NyQuil has Tylenol in it, so they will tell you over and over again, no, I didn't take Tylenol. Not realizing that, well, yeah, when you took that NyQuil, you took Tylenol, right? So it can be very powerful to have sort of all of those molecules in terms of the coverage as well.
Excellent. And nice segue to the next question regarding clinical trials. During clinical trial design, it may become necessary to instruct patients to follow a specific diet in order to be effective in responding to the treatment. Is this a reasonable assumption?
Yeah, I think you're right. I think the previous was a nice segue. Clinical trials, the idea of diet. So certainly we know that, and well, what we've discovered is that certain dietary signatures, the famous one being grapefruit juice in treatment of, what is it, blood pressure? Blood pressure medicine? Anyways, that the grapefruit juice can have influence on either making a drug more available or less available in some people, leading to some toxic exposures in certain individuals. And so there was this profound impact of diet in that model. So certainly, first you have to understand if that's out there, but we know that that can have an impact. So diet can also lead to quite a bit of diversity within a population, right? I refer to humans as being free-range humans, right? We're out there, we're eating whatever we want, we're taking whatever medication we want. As much of that variability as you can remove from the system, the better. Now, that does bring questions in of whether people are actually following the dictates of that diet. So that will always be a question. But I think there's some helpful things that would come out from a controlled diet within a clinical study. But if you can't do that, that doesn't mean you can't find really interesting things. It just means you probably have to power your study a little bit higher to make sure you can get through, a little bit of noise that's introduced as a result of the variation in diet in the population.
Perfect. Another question for Kirk. You mentioned all these microbiome metabolites. Can you comment more specifically about how your technology is being used to understand host microbiome interactions?
Yeah, I mean, hopefully the examples illustrated that, and just imagine that across any other kind of biological question where it's suspected that the microbiome is playing an important role. Samples can be plasma, they can be urine, they can be fecal samples, but I think the main point here, I think in this webinar regarding how our technology is helping, is really how comprehensive the platform is. Being able to see such a wide variety of metabolites, endogenous metabolites, or host metabolism, as well as a lot of microbial metabolism, as well as the xenobiotics that come into our body and are actually have a whole life history of ADME, including the microbiome. So it's really coverage and then the ability to interpret that data.
Perfect. And we do have another diet-related question. Can you speak to the concept of the keto diet in patients who may also take high doses of glutamate as a supplement in order to heal the leaky gut?
Kirk, do you want to address that one? I don't know if you think that would be related to the ketogenic diet. Yeah.
Yeah, I'm not really that familiar with all the treatment regimens that these patients are under, so I can't really comment specifically on that.
But what I would add is that we'd be able.
Oh, go ahead.
I was going to say, what I would add is that we'd be able to detect these changes in the diet, so that if someone's on a ketogenic or glutamate-rich diet, that's something that we can confirm because we can look at compliance with our platform. So making sure that the diets, as Annie was saying, that the diets that people are on, they are sticking to them. We can help ensure that. And then the biology has to be informed by the outcomes. So this falls on the investigator then to associate these data with specific outcomes to identify the biology behind these.
Our next question is for Greg. Can samples across different cohorts be compared directly?
Yeah, so I believe that speaks to what Annie was talking about, one of the centerpieces of Annie's talk, of the comparability. Because we have spent so much effort and intellectual investment of developing our platform to be able to compare samples using standardized QC samples that are included on every run and every study, we can directly compare across different cohorts, across different populations in direct ways, which is one of the powers of why metabolomics is really at the forefront, or should be at the forefront of the omics stream of technology.
Thank you. We have time for just one more question. This one is for Annie. For endocrine health, the small window doesn't give the full picture. Large targeted panels are necessary of 50+ compounds. How do you address comparability and QC for these large panels?
Yeah, I mean, you've really hit a big question here. As we move metabolomics into more of a precision medicine space, right, as we get closer to the sort of the patient bedside, if I'm understanding your question, it becomes really important that the metabolomics, the information in metabolomics is incredibly helpful at really getting a much better holistic picture. And that's why these panels have these much larger number of molecules, because that's what we find that you need to really understand what's going on. And QC and QA is really what distinguishes metabolomics technologies as you move from sort of traditional RUO, research-based stuff, research projects only, towards, moving this into a more clinical space. So Metabolon is actually CAP certified, so the Clinical Association of Pathologists come in, they audit us yearly. And we're also about halfway through our ISO certification for the EU. And basically that just means that we have a defined set of procedures, and a tremendous amount of quality assurance and quality control samples that go into each batch of samples that we run to allow us to count for things like instrument sensitivity changes over time. And this is really what's enabled us to do the case study I talked about with the Baylor, where we compare an infant that we ran, that we just ran that sample today, but we actually have a reference healthy cohort that we ran over 5 years ago, right? So we can actually compare that patient today to those samples that were run 5 years ago. The only way to do that is to have a really defined quality control process where you have confirmed QC samples that you can say, hey, am I diagnosing the same set of diseases out of this confirmed QC sample? Yes, okay, everything is where it should be on the platform. And it does take a tremendous amount of, just extra samples being run in addition to your experimental samples to be able to meet those sort of needs. Hopefully that sort of got to your question.
Thank you very much. We have another full 2 pages of questions, but we've come to the end of our time. So I'd like to thank our presenters today for their presentations. I'd like to thank Metabolon for sponsoring today's event. And finally, I'd like to thank those of you who came and spent this time with us. We know you're very busy and that you could have been anywhere else other than with us. So thank you for choosing to be here. We look forward to seeing you at future Cambridge Health Tech Institute Web Symposia. Thank you all so much and have a great day.